I might speak for a bit less time than planned, which is probably a great relief for most people. Really carrying on the same themes to look at carrying these genetic loci into images and doing voxel-based associations and so forth. I'll spend maybe 5 minutes at the beginning talking about Enigma, which is an imaging genetics consortium, just talking about how that's organized and how it came about. So most of the talks so far have focused on trying to understand how genomic variation is associated with different types of features you can compute from biobanks of images. So you could look at MRI, you could certainly look at functional data, as Jason mentioned briefly, or you could look at molecular patterns in either spectroscopy or histology. It just so happens that the anatomical data is the most plentiful so far, so perhaps in the coming years of brain mapping, we'll see a shift down into some other types of modalities as well. So many of us have been using different biobanks to look at genetic effects on the brain. A little under 10 years ago, Enigma began to publish some of the largest worldwide studies that combine imaging and genetic data, ultimately with the goal of understanding factors in the genome that are influential on the brain, either in a helpful or harmful way. And right now, there's a little over 60,000 MRIs with associated GWAS data that have been compiled across the world from a little over 300 medical centers. Now, in tandem, there have also been very large-scale studies without genetics, just looking at what are the differences in these different disorders, certainly with MRI. And you'll see some diseases listed here where Enigma's been publishing some of the largest studies of the disorders. So I think, as other speakers have mentioned, one of the benefits of large-scale collaboration in imaging genetics is, first of all, statistical power. You can identify smaller effects than have in the past been identifiable with smaller samples. Also efficiency. I mean, if there are more and more biobanks, ultimately there'll be more data than each person can identify on their own, so the distribution of the computation is helpful. And also team science. I think most of us would agree when you work with a lot of other people, a lot more ideas come into play, certainly in terms of interpretation and also in terms of innovation. So obviously these are very, very large-scale studies. I just want to point out that some of them are public data sets, some of them are being collected. For data that is private, obviously it's not always possible to send genomes around, certainly with the new data protection laws. A lot of the data stays at the site where it's being collected. So that's why you'll see a lot of studies are meta-analyses. They basically have independent analytic teams at different sites, and they come together at the end to pool their evidence for a particular genetic locus being implicated in the disease. So just really wrapping up talking about Enigma, there are studies of brain diseases, 18 different brain diseases looking at how they affect the brain, but you certainly have enough power to look at genetic association, all of the letters or nucleotides in the genetic code, to discover genes that affect brain aging or development or function or disease risk. Now, Enigma is organized into different working groups. There are groups that study the different disorders that Sven mentioned, schizophrenia, bipolar, MDD, but many working groups in Enigma grew up because there were not really enough highly powered studies of these different disorders. So certainly in autism or OCD or ADHD for that matter, a lot of the studies have smaller effect sizes than you'd see, for example, in epilepsy or ataxia, some of the neurological diseases. So clubbing together, it's been possible to pool images and genetic data on these disorders and get better studies. And also Enigma has studies that, working groups that look in the genome, not just GWAS, but also Carrie Bearden's going to talk after the break about copy number variation. Epigenetics is also an important source of variation in the genome. Jason and others lead a group on evolution. So I won't talk about all of these, but we'll talk about some of the major highlights. So because the data comes from all over the world, obviously there's a lot of interest in whether findings generalize from, I think Sven mentioned this at the end, primarily, for example, European studies through to Asian and other data sets in different parts of the world. And so this diversity of data gives a little bit of an ability to triangulate not just the genetic data, but also the imaging data. There's a nice review by Carrie Bearden, who will speak after the break, on the relevance of these types of data sets to genetics. So let's get going with common genetic variants. So I think, as many other speakers have mentioned, about 10 years ago, a common genetic imaging study design would be to say, I was reading in the news that this new gene is implicated, for example, in Alzheimer's disease. In fact, this was one, TREM2, is a variant where the Alzheimer conferring allele is found in about 1% of people. So it's either a common variant or a rare variant, depending on your definition. But this was very interesting because a lot of drug companies want to find genetic variants that speed up brain aging. And so in this case, It was associated with a faster loss of brain tissue in longitudinal MRI. And so this was published, and it was based on the ADNI data set. This is about 5 years ago. But the problem with this, and I'm going to criticize it because it's our own study, is how do we know this will ever be found ever again? I mean, if a drug company designs a drug trial and they enroll the 1% of people that have this gene, do we really believe that they're going to lose brain tissue faster? And I think if we had a survey, most of us are sort of seasoned imaging genetics folks, we might say, oh, I don't really know if this is going to hold up. So one of the reasons for doing imaging genetic meta-analysis in multiple cohorts was really to verify whether these effects hold true and find others. So the skeptic side of the debate was maybe championed by some of the psychiatric geneticists. Here's Nick Martin. He was the president of the Behavioral Genetics Association for many years. And he said that if you want to discover genetic loci that affect the brain, you'll need brain images from 50,000 people, just because images are more expensive to collect than measures of height or weight. It doesn't change the power calculation. So let's call that Nick's bet. And it was a slight simplification of his position. But maybe the default hypothesis is this isn't going to be very efficient at all. So I think, as Jason and Sven mentioned, the design that you use to look at statistical variation in the genome and its effect on images resembles the design that you would use in linking genetic variation with a disease. So borrowing the analytic methods from the Psychiatric Genomics Consortium and other genetics consortia, you look at the genome, try and identify signals that are associated with that genetic variation. And a lot of the confidence that imaging was going to be more efficient came because either the things that you measure in images are more proximal to the mechanism of gene action, or they're more precisely measured. And so again, this may give us a door to finding genes implicated in disease more efficiently than in the past. Now, I'll tell you a little bit about one of the earlier studies that was done by Enigma, borrowing this methodology of genome-wide association and applying it, in this instance, just to the volume of different parts of the brain. So you can imagine having a whole lot of MRI scans running FreeSurfer, doing a lot of QC, and asking the question, where is it in the genome that there's evidence that there's common genetic variation that is associated with these volumes? And that's just a summary. As the earlier speakers said, these are Manhattan plots. On the x-axis is a map of the genome. It's about 1 million variants that could be associated with the volumes of these parts of the brain. The y-axis is the evidence, and so it's the negative log base 10 of the p-value. And that little sort of high jump bar that you see dotted there is the 5 times 10 to the minus 8 p-value threshold, which essentially is 0.05 divided by 1 million, because you're doing about 1 million tests. You can sort of have different opinions on whether that's the right thing to do, but in genetics, that's really the most conservative way to proceed. So just a little bit on the mathematics. Genotyping chips do differ if you're going to do a multi-site study and people haven't genotyped using the same chip. You can impute the genetic variation using common reference data, 1,000 genomes. It used to be HapMap3. And so you get a set of common loci, just like you get a set of common pixels in the image, a set of common loci on the genome. And you're essentially doing multivariate, linear regression at each locus on the genome, trying to predict the trait. And you tend to remove confounds of no interest, like age, sex, age squared, things that you don't think are going to be interesting, and you want to get them out of the way before looking at the genome. So I think, as Sven mentioned, you could say, well, I wonder what this is going to look like. Either it'll look like the genetics of Alzheimer's disease, where there's one astronomical source of risk in the genome. So if you look at ApoE, this wasn't a genetic variation that was discovered through GWAS. It was discovered even longer ago through linkage. But about a quarter of us carry the ApoE4 genetic variant. It triples your risk. If you're unfortunate enough to carry 2 copies, your risk goes up 9 to 15 times. So this is the exception rather than the rule. It's a really dangerous gene. Most of the others that have come up as associated with Alzheimer's disease in the recent past change your risk by about 10 to 20%. But that's kind of useful. So as we heard prior speakers mention, you could prorate from this paper a polygenic risk score, which sort of weights your ApoE, these other 20 loci that affect your risk by about 10 to 20%, and come up with an aggregate score or polygenic risk score. And one of the ways of doing this is just to count the risk alleles, which is a little bit simplistic. Another way is to weight them by their odds ratio, so the ApoE gets a lot of weight, the others get less weight. And you could do this at different thresholds. You could include a lot of loci, you could include only a few. And it's extremely useful. So this is a paper by Rahul Desikan and colleagues, where, I mean, if you just wanted to look at your overall risk of Alzheimer's disease throughout life, the two dotted lines are whether or not you have an ApoE4 haplotype, and a quarter of us have that, 3/4 of us don't. The x-axis is how old you are, and the y-axis is how likely it is every year that you'll be developing Alzheimer's disease. And so, obviously, until your 70s or so, for most of us, luckily, that's about 1%. But you can see there's just absolutely astronomical risk later in life, particularly if you have a high polygenic risk. And I think these different curves, which are different quantiles of polygenic risk, show that it's not all ApoE. Some of these other loci are quite interesting. And if it makes this much difference to health in the latter part of life, then it's extremely important to try and shut this off or interfere with it, or at least understanding in the imaging what types of processes these genes are causing. Now, Sven mentioned the schizophrenia GWAS and bipolar and major depression are similar. They look a little different in that the distribution of genetic risk in the genome, in other words, the common loci that are associated with higher risk of, in this case, schizophrenia, they're much more spread out. There are no really big hits. There's no sort of one risk gene that's the best. And I think, as Sven mentioned, this was only really appreciated once the sample sizes for these studies became truly vast. I mean, there's now like 250 loci that are associated with heightened schizophrenia risk. So obviously, we'd like to know if any of these things have an impact on the brain. So you could ignore all of the disease studies and you'd say, well, look, we're just going to copy them. We're going to run FreeSurfer on the MRIs. We're going to run TBSS on the DTIs. We're going to try and do that in a harmonized way. We'll look at the differences in acquisition protocol and make sure that isn't influencing the answer. But we're going to run linear regression at 1 million SNPs. Obviously, sample sizes vary. So we're going to take a meta-analysis approach. There are other approaches, but one approach is to combine the effects across sites in such a way that each site's vote in the overall outcome depends on the sample size. And you can use other weightings. You can use the precision of the measures and other types of things. But in the end, this makes sure that you can find effects that no single site can pick up on their own. And if you have a site of 100 scans versus UK Biobank with 15,000, each vote is counted in a sort of roughly proportionate way to its contribution. Now, this certainly works, and I think this is a study that Jason and many others worked on many years ago. You can find, if you look at just the size of the hippocampus, loci in the genome that are associated with the volume of the hippocampus, carrying the major versus minor allele at the top hit. How much difference does it make? It seems to make your brain look about, on average, 6 years older in terms of, 3 years older in terms of hippocampal aging. So it's not a vast amount, but it's a fair amount. And then you can go back into the image, and because in imaging we can do statistical parametric mapping, you can say, now that I've found a locus that appears to influence the volume of the structure, can you map, is it all of it? Is it particular CA fields of the hippocampus? What is the spatial scope of effects? And this can be iterative. You can go between the image and the genome and refine the loci in the genome that you think are influential. And again, these studies do tend to get more and more well-powered. I think most of the surprise, as earlier speakers mentioned, was that you really need about 8,000 to 10,000 people to overcome this multiple comparisons correction of doing 1 million tests. But once you've gone beyond that, there really are robust findings. Now, in terms of figuring out what these loci mean, I mean, you can certainly look at the locus and say, well, this is near the harakiri, apoptotic cell death gene. But unless you look in patient populations that carry versus don't carry the allele, it's difficult to ascribe a function. And this was a study, very interesting study by a Russian geneticist who noticed that the top hippocampal variant was also carried in different degrees in a pedigree she was studying in Russia, and was associated with mental retardation and psychosis. And as a case of serendipity in science, she was doing linkage studies in ethnic isolates and found the same region of the genome. It was very, very interesting convergence. Now, there's some limitations with genome-wide meta-analysis, and here's a very conspicuous one. ApoE, which we just said was one of the most dangerous genes in the genome, it doesn't operate uniformly throughout life. I mean, you can be a young adult and not have any impact of carrying E4 yet, whereas in your 60s and 70s it could be horrible. And so in Enigma, we try to find out, does carrying the ApoE4 genotype that confers triple your risk of Alzheimer's, does it shift your hippocampal volume in cohorts that have a mean age of 20 or 40, or for example ADNI, mean age of about 60 or 70. And it turns out that your z-score for hippocampal volume is shifted by about 2 standard deviations if you carry E4 late in life, but hardly anything at all if you carry E4 early in life. And it may very well be that many genes are like this, and if there's an age-dependent effect, you wouldn't necessarily find it by averaging together all the cohorts that you have. Now, I have to say, the Enigma discoveries tend not to be like this. They do tend to discover loci that are consistent in their effects throughout life, but of course we would miss them if they weren't. And so it's sort of an important caveat. Among the variants that seem to be associated with, for example, the total size of the brain, intracranial volume, there are some quite famous genes. And so one of the ones, MAPT, microtubule-associated protein gene, that's a gene that comes up in all sorts of GWAS for Parkinson's disease, CJD, other types of degenerative disease. People with an allele in this gene that confers disease risk also have alterations in total intracranial volume. And of course, these studies have been really undertaken by lots of institutions phenotyping their images together. So about 2 years ago, this was the sort of lay of the land in terms of the number of hits. It didn't look like it was much more efficient looking at hippocampus or total volume of the brain versus the other genome-wide screens. And this line of work has carried on. So if you look on bioRxiv, there's a beautiful study, 40,000 MRIs, with 45 variants in the genome that influence the volume of one of the subcortical structures at least. And I think there's a great deal of interest in what these do. I mean, we don't really know yet whether these influence disease risk. But the early evidence is that there is, as Jason and Sven mentioned, some overlap between the loci that are associated with differences in brain volumetry and disease risk, particularly for Alzheimer's, but also for bipolar. Not much evidence for schizophrenia. But there also seems to be an implication of the parts of the brain involved in those disorders as well in the loci that are being affected. And these are, again, very, very large-scale studies, early days yet, understanding what the loci do. And of course, the sources of information could be very varied. You could look, as Jason and others are looking at the evolutionary development of the loci that affect brain volumes, or in the paper on the bottom, whether in infants the genetic loci already have an effect. Or you could do informatics with these. So there's a browser online called Enigma Viz. If you want to look at different parts of the brain and the genetic loci that influence them, you can look them up. Or if you have a gene that you're interested in, you can look up what parts of the brain they affect. And I think, as with anything where new data is coming out, I think very, very early days trying to understand what these do. But there are some principles that seem to occur. So if you go from one region of the brain to the next, you could either say, well, I think all of the same loci will affect all the brain. I don't think there will be any regional specificity. Or you could say each gyrus or each functional area might have different genetic architecture. And it's a mix of both. So as you move gyrus to gyrus, there are some common genetic variants, some new ones, and it looks like there's a series of perhaps partly overlapping modules in the cortex that have similar genetic determination. And one of the nice things about this is that the p-values are so strong that you could begin to look at statistical parametric maps without that additional correction for image-wide search costing you a very heavy penalty. Now, just a couple of terms. If you look at different parts of the cortex, either the surface area or the thickness, and you look at the p-values for the genetic associations, you can try and understand, is there any pleiotropy? So do genetic loci that influence the thickness of a part of the cortex, are they exactly the same ones that affect its surface area? And there's a little bit of overlap, but not universally. And I think one of the surprises is that thickness, which has been so dramatically implicated in nearly every disease we study, it doesn't seem to have quite as many genetic hits as surface area. We don't really know why that is. But if you lay out the Manhattan plots, on one axis there's the genome, on the other axis there's the parts of the brain, you can begin to see what the lay of the land is for looking for modules, looking for particular hotspots in the genome with different gene categories that might be particularly implicated. And I don't know, it's hard to understand what's going on here. But one of the ways into this type of data where you have brain regions on one axis and genetic regions on the other is to see if there are any patterns. And so are there, in the loci, in this case, that influence cortical measures, are there hotspots on the genome? There are some, but it seems that the genetic determination is spread quite uniformly over the genome. Is this consistent from one cohort to another? So if you look across Enigma at all the cohorts that have taken part, some of the loci that affect the volume or the area of certain parts of the cortex, they're supported in, for example, 35 of 36 cohorts. So there's just overwhelming evidence. And 2 days ago, this was a total of 51,000 MRIs in the study. And just zooming in, I mean, these are cohorts as diverse as ADNI, which is elderly people, EpiGen, which is the study of epilepsy, LBC, the Lothian birth cohort in Scotland, many, many different cohorts here. And you can see the degree of agreement. These are the error bars on the association of a particular locus, and in this case, the surface area of the precentral gyrus. And you can see for the first time in imaging genetics, or perhaps one of the first times, the vast majority of the cohorts are supporting an effect rather than the vast majority being null. Now, you could say, well, is there a more sophisticated way of understanding the effects of the loci? It looks like there's some initial evidence that the loci that are associated with smaller brain regions in some areas are associated with larger brain regions in surrounding areas. There may be some sort of conservation principle. I think Jason alluded to this earlier. If you sort of expand one area of the cortex, you might shrink another area of the cortex. And certainly during corticogenesis, there is some evidence that the migration of progenitor cells, there may be some sort of conservation principle where certain genetic loci promote the migration of cells to some areas versus others, or there might be CAM kinases or certain axon guidance genes that steer these cells to the right place or to different places. So this is very, very testable and interesting hypothesis. The other question, going back from the cellular level up to the sort of behavioral level, is do any of the genetic variants that are seen to affect cortical, for example, morphometry, they also affect IQ? They do. The loci that tend to increase cortical volume or area and thickness, they also tend to be associated with higher IQ or higher education level. There's some surprises. I mean, there are loci that have been implicated in disorders such as insomnia. You could ask the question, do the insomnia risk genes affect any particular parts of the brain? It looks like these are some interesting regions. And you can go through some different disorders and say, for the loci that I think might be involved in a certain disease, are they some of the same ones that are involved in a particular region of the cortex? So that's very, very much ongoing work and also interesting. Now, you'd also say, I don't really care much about this. It was a functional brain mapping meeting. Can you show us some signs that these loci affect function? Now, I have to be honest, the reason there have not been quite so many functional studies is that MRI data, structural MRI data, is more plentiful. So you can certainly aggregate in Biobank 20,000 MRIs across Enigma, over 50,000. But this is a sort of early days of interesting study. Dirk Smit, looking at EEG. I mean, those of you interested in EEG, you can look at all sorts of things like power in different frequency bands to see if some of the variants that we've implicated in either cortical structure or disease are also influencing the functional synchrony of the brain measured by EEG. And this is sort of an intermediate phenotype. I mean, there may be an aberrant pattern of activity that is quite identifiable using EEG, but also has a common genetic variant profile that affects it. And there are some interesting variants here. So GABA, major inhibitory neurotransmitter in the brain, there are loci implicated in alcoholism, epilepsy, other disorders of inhibitory neurotransmission that appear to be associated with differences, in this case, in 20 hertz oscillatory activity in the brain. And it may very well be that a combination of structural imaging, EEG, and other markers may help us understand if these loci affect functional brain synchrony. This is an interesting way forward. Now, you could say, well, I don't think you're doing this right. I think one of the reasons that this is so inefficient is that you have this sort of jigsaw model of the brain, and you haven't broken it up properly. And if you did a better job, particularly with voxel-based mapping, this might be more efficient. So you could basically, now that the hits are so highly significant, run a genetic association at each pixel in the scan. So this could be voxel-based morphometry, it could be gray matter density, it could be tensor-based morphometry. And you could say, tell me as you go through the genome if there are any loci that are particularly predictive of the signal in this voxel. And so long as you do some nonlinear image registration of the cohort data from one site to another, you could get different hits. And this actually kind of works. So if you look in, for example, ADNI, which is a North American study of Alzheimer's disease, the top locus in these genome-wide screens actually affects the volume of the putamen. It doesn't look like it has a very clear pattern. If you look in ADNI 2, which is a sequel to ADNI, it doesn't look like it has a very clear pattern. HUNT, which is a Norwegian cohort study. It's kind of similar. QTIM is an Australian study. But by meta-analyzing all of these different voxel-based maps, you actually get a quite localized genomic effect. And it may well be that voxel-based maps give us these sort of modular or regional effects that we might overlook if we were looking at these things in different ways. So just to sort of cover what's going on there, you could certainly do voxel-wise analysis, in this case of structure, but also a function, where many noisy maps are pulled together to really find a quite circumscribed effect of a genetic variant. Now, Jason showed this earlier, and I have to say it's a little bit of a controversial topic. So even though the SNPs so far discovered that relate to brain structure, with the exception of ApoE, which is a very unusual one, even though they only explain less than 1% of the variants, we do know that if we discover them all, as these heritability estimates go forward, you'll be able to account for between a fifth and a third of the variants in brain structure. So even if you don't care about genetics at all, it's extremely interesting to remove this. I mean, if you could reduce about a third or a fifth of variants in your study, that would be very worthwhile. But a lot of the controversy, I think, has come from trying to understand whether imaging markers are any more explanatory of variants than the others. And there's been a very interesting recent development in this. If you look at this paper by Dominic Holland at UCSD working with Anders Dale, he looked at all of the GWAS that have been done of imaging data sets of psychiatric and degenerative disease and said, on the x-axis, if your sample is 1,000, 10 to the 3, or 10,000, or 100,000, how much of the genetic variance is explained? I mean, are some of these phenotypes just particularly efficient? Are some of them a nightmare where you'd have to phenotype 1 million people? And they argue in this paper that the discoverability of genetic loci that affect different traits is orders of magnitude different. And so, going back to Nick's bet, it seems like there's some evidence that certainly the structural phenotypes have a simpler and less polygenic architecture in some cases. But I think, as Sven mentioned, things like major depression, which have multiple causes, multiple etiologies. And so this is really quite a nice plot that there might be on the x-axis 1 million people needed to find half of the genetic variants involved in, for example, educational attainment, whereas by the time you phenotype 100,000 people, you have nearly all of the genetic variation in brain volumetrics, in this case for the putamen. Now, another line of skepticism is that this is really just the wrong way to be doing it. So it's been traditional in imaging genetics to do mass univariate testing. In other words, you say, tell me the SNP that has the best predictive value in the genome. And I mean, we just showed voxel-wise screens. We're doing the same thing there. If you fed textbooks into a machine learning algorithm that were in, for example, French and English, there wouldn't be a certain letter that was different. I mean, it's a much higher order syntax that distinguishes languages than this sort of single letter variation. So if you were to come up with a more mechanistic syntactic model of how genes act, you may be much, much more efficient. So the first pass of doing this is something like multivariate imaging genetics, where you say, I don't really think that there are single loci that explain much of the variance, but if you look at the genome on the right there, let's do a polygenic risk. We'll do a linear combination of genetic loci. And in the image, we can also do a linear combination of values. And those of you familiar with CCA or PLS, they're simultaneous diagonalization approaches in which you project the data you have in the genome and you project the data you have in the image on a vector. And when you've picked the best vector, and best in the sense that the two measures are maximally correlated, you found a linkage that you might overlook if you were doing this pixel by pixel or locus by locus. Now, it's worth thinking of some toy examples. It's certainly possible that you could never find a locus that is useful, but you could find completely perfectly explanatory maps in the image and polygenic risk profiles in the genome in that case. And so there's a very interesting paper by Marco Lorenzi at University College London about how you would do this projection approach at multiple sites where the genome data remains private, and you send around vectors, and they're a little bit like keys. You project your genetic data onto this sort of private key, and then keep passing it around to get better efficiency. The third source of skepticism is that really we should be looking at other types of genetic variation. So Harry Bearden's going to talk about CNVs. You can certainly look at epigenetics, the methylation of the genome. You can look at the genetics of brain change over time rather than structure at any one time point. And some of the initial forays into looking at epigenetics in Enigma have been quite interesting. You can certainly look at the methylation status of genetic loci. They're not the same ones that you look at in GWAS. The CPG sites that tend to be differentially methylated. And Sylvain de Rivière in London, who's beginning to find loci at a genome-wide level in this study, she's finding places in the genome where a higher degree of methylation, not the SNP, but the methylation level, is associated in this case with hippocampal volume. So that's a very nice new direction for the future. So in the remaining couple of minutes, we won't be talking about any of this at all. Maybe another day we'll talk about this. But maybe I'll just show one slide about this. There is, at the same time in Enigma, a very concerted effort to look at the distribution of brain differences in cohorts across the world. And it may very well be, as we were arguing earlier, that the linear combination, or could be nonlinear combination derived from machine learning, of these regions or phenotypes may be much better correlated with genetic variation than the markers that we choose today, a sort of piecemeal approach of one region at a time, maybe just simply overlooking the signals that matter the most. The other sort of fourth line of skepticism is that the phenotypes are terrible. So, structural morphometry, if you're looking at something like anxiety or something that isn't necessarily imprinted on the brain structurally, you might be better off looking at, in this case, at the connectivity or the functional synchrony and resting state. And now that the significance of the genetic variant associations are so high, you can basically pack this type of data into connectomes. You can certainly do genome-wide associations at each element of a connectivity matrix. And it used to be thought, at least comparatively recently, that you would just blow your chances by looking at so many data points. The multiple comparison correction in the connectome would be too prohibitive. But I think now there's certainly loci in the genome that have very high significance, and you could test them across these arrays. This is just an example. You could sort of find tracts, or it could be functional synchrony that you're surveying for common variant effects. And these may be much more fruitful than the measures that we've been looking at so far. So just to wrap up, we're not going to talk about any of that. Just a couple of review questions. So Jason said, what should we think about in doing these studies? One of the things to bear in mind is, why is it that GWAS, or genome-wide association, has tended to be used in favor of candidate gene studies? I think the main reason is that the replication rate was just awful. A lot of people have thought that these effects would be large, and they're not. So it's not very surprising that a study with, say, 100 people that's looking at an effect that's expected to be under 1% will not replicate it. The other question that's been a bit fractious is whether the polygenicity or genetic architecture of the imaging traits is simpler than those for example, psychiatric diagnoses. I don't think we can answer it. And so at one end of the continuum are the people that say it's no different. But there are certainly, as we've gone through in the talk, traits or ways of combining traits or more direct measures of brain function that are likely to be more efficient. But I mean, that's a bit of a cop-out. We've got to say what they are. But this is still an open question. And then obviously, finally, GWAS is not without its critics. It's very interesting, and you certainly find genetic variants that are very useful. But it's important, I think, to bear in mind other types of genetic variation. And Carrie Byrne talked about CNVs. We talked a little bit about methylation and epigenetic variation. And it may very well be that in 5-10 years' time, we are looking at very different types of genetic variation than we're looking at today that may be more fruitful. So that's it. Thanks for your interest. Thanks very much.